This is Agnes, the presenter of Sickle-Cell Talks with Agnes. Welcome to my YouTube channel. So stay tuned because every week we'll be posting videos about any topic in sickle cell disease. We don't have set days yet, but just turn on the post notifications for you to be notified every time we post a video. Thank you and stay tuned. If you've got any questions, get in touch with me on social media or social media platforms. Thank you.
Thank you so much, Agnes. So I'm going to share my screen. We all practiced this yesterday. I've got my talk ready. So I just do that and do that, I think, and share. And hopefully you can see a picture of the Sydney Harbour Bridge. So I'm here. The Indigenous owners of the land where I am in Sydney's inner west are the Gadigal people. And they lived on these lands. But I can tell you today it's cold and wet in Sydney. So I'm going to talk about, it's 1:15. I'll talk about for 25 minutes about the work. Now, So I want to tell you about this long road to better therapies for sickle cell. I'm sorry it's taking for so long. Scientists have been interested in this topic for a long time, but it's turned out to be harder. I put that picture of an obstacle course. It would be easy to do that obstacle course as an adult, but in the world of understanding molecular genetics, we're like infants, and it's proven to be very hard, but we are getting closer. And what I want to explain to you this evening is the pathway towards a cure. I want to give you a sense of the history, some of the people and the path, and to show you what has been happening and is happening across the world, and also to explain what's happening in Australia. So many people, I've heard sickle cell being called a rare disease or not a well-known disease, but it is actually to scientists. And I point out, as Agnes said, I'm a scientist, not a clinician, but to scientists, it's the most famous inherited disease of all. And it's become very famous because it has been a topic that some wonderful scientists have worked on. And I've shown some of this on this slide, and it gives you the impression, you can even tell from the black and white photographs, how people have worked on this for many years. Linus Pauling, who was unusual in winning 2 Nobel Prizes, one for chemistry and one for peace. He was the first person to describe sickle cell as a molecular disease. Max Perutz and John Kendrew solved the structure of hemoglobin, the molecule which is mutated in sickle cell, and they won the Nobel Prize. Fred Sanger, his sequencing method was used to sequence the DNA change. You'll know it's the disease, inherited diseases caused by one DNA change. He won the Nobel Prize. Janet Watson, she worked in New York, and she made an absolutely important observation about fetal hemoglobin, which is a different type of globin that you have when you're a baby in your mother's womb, and it has high binding to oxygen and allows the baby to take the oxygen out of the mother's blood into the baby's body. And she found out that some people have more of this fetal hemoglobin and they have milder effects of sickle cell. I'll come back to that point. The next picture is Francis Collins, who led the Human Genome Project, and he worked on sickle cell. Andy Fire and Craig Mello won the Nobel Prize for turning genes off, and their work central to the story tonight. Stu Orkin is next. This year, he won the King Faisal Prize, which is one of the most prestigious prizes in science, and he spent his life working on sickle cell. I'll tell you about his work. And then we have Emmanuel Charpentier, Virginia Siksnus, and Jennifer Doudna. And Emmanuel Charpentier is an inventor of CRISPR, and their first clinical trial is on sickle cell. So on the next slide, I just show that the people in red there have won Nobel Prizes. So work on hemoglobins and on sickle cell has been among the most important work in the world. And it started so long ago. If you see the picture on the right, that's from George Gulliver's book, where he went around, he was a retired soldier. In 1846, he went around London Zoo, collected blood from different animals and looked at it under the microscope. And the blood cells are so different, very, very different. Different types of animal have very different blood. And he was the first who observed sickle-shaped blood cells in deer. And deer don't suffer diseases from that, but those were the first sickle cells ever seen. But then the history goes further. In 1910, so that's 110 years ago, James Herrick of Chicago observed the first sickle cells in a blood film on a glass slide of a medical student who was studying there in Chicago, a medical student from Granada who had sickle cell disease. It was 1949, Linus Pauling realized it was a molecular disease due to a single change in a single gene, changing the protein hemoglobin, which carries oxygen around the blood. Another nearly 10 years go by, 8 years go by, and Vernon Ingram defines the exact change in the hemoglobin protein. A few more years, Max Perutz and John Kendrew solved the exact structure of hemoglobin. Fred Sanger's sequencing, another nearly 20 years later, identifies the precise mutation, and Fred Sanger won the Nobel Prize. He also won 2 Nobel Prizes. And then just a couple of years ago, the geneticists, now that human genetics with the human genome has become so important, worked out that this mutation arose 260 generations ago, 7,000 years ago in Africa. And if you had 1 copy of the sickle cell gene, you had less severe malaria, but 2 copies and you had sickle cell disorder. So all of this work over this time was extremely important, but it didn't help us to provide a cure. But the clinical observations did suggest treatments. So I mentioned Janet Watson. She noticed that children had fewer symptoms than adults, and she attributed this to the fact that children still have a little bit of fetal hemoglobin. That's that high affinity globin that binds oxygen tightly that's found in babies and allows the baby to take the oxygen from the mother's blood. And this observation, she was extraordinary that she worked this out. It changed the whole direction of research. And so that's why I've said, I've got in this slide, yes, humans have different globin genes, those active in utero, which bind oxygen tightly, and those turned on later. And some people have slightly higher levels of fetal hemoglobin throughout their whole life, and they have mild sickle cell symptoms. And so scientists said, well, if we could turn on this fetal hemoglobin gene, or even just increase its expression a bit, that would be a treatment for the disease. And George Stamatoyannopoulos, he and his wife Thalia came from Greece, where there is much thalassemia, a disease like sickle cell. And he dedicated his life to raising funds and doing research. He was a great scientist on sickle cell, and he had a conference where everyone in the field goes every 2 years. And people discovered drugs that do increase the level of this fetal hemoglobin, this superglobin. A DNA demethylating agent, 5-azacitidine, works, but it is associated with changes in DNA and could cause cancer, so it's never been used in humans. But other drugs, called cytotoxic drugs, which change the blood stem cells, and hydroxyurea is one, that does turn on fetal globin, and that's widely used. It is under medical supervision. It's a safe drug to use. All drugs can have side effects, but it's a recommended treatment, and I believe it's used widely. But no one knows how these drugs work, and they don't work equally well on all patients. And because we don't know how they work, we don't know how to make them better. So those were great. That was a great drug developed in the 80s, but it hasn't really been improved on dramatically. So scientists went back to the bench and just said, we're going to learn everything we possibly can about hemoglobin. They studied the genes. Any of you who've studied biology might have learned about promoters and enhancers, the control regions of genes. You might have learned about polya sites, the endpoints of genes. These were all characterized studying the hemoglobin genes. If you've done a lot of molecular biology, you've learned about histone modifications, hypersensitive site, master regulators, pseudogenes, splice site mutations. All was discovered in the globins. So the globins was a tremendously exciting area to work with. But all this knowledge we hoped would provide a cure, but it didn't provide a cure. And for 30 years or so, molecular biologists worked, but we weren't closer to a cure. Then this wonderful woman in London, now works in NIH in Washington, Swile Thane, took a different approach. She realized the human genome had been sequenced and many people were having their DNA sequenced, and you could analyze many different families. And she said, we know that some people have more fetal globin expressed. I wonder if there are particular DNA variants which correlate or cause extra fetal globin. And she found one, BC11A. Changes in BC11A increased fetal globin. And my former supervisor in Harvard mutated the BC11A gene in a mouse, and fetal globin was suddenly turned on. And this was a wonderful moment. This was a eureka moment. People realized if you can inactivate BC11A, you can turn the fetal globin on. So if we can change the BC11A gene, or we can find a drug, that could be a cure. And this was only less than 10 years ago. The whole field shifted pace, and we said, now we know BC11A is the answer. It sits on the fetal globin gene and turns it off. I'll explain that in a minute. And we all switched to trying to turn the BC11A gene off. And you might be thinking, well, why didn't we just turn the fetal globin gene on? It's so much easier in biology to break something and turn it off than to turn something on. So I say, if me or anyone here wanted to work on my car or bike, it'd be easy to make it go slower, very hard to make it go faster. But now we had a target which we just had to knock out. And Stu Hawkins' lab in Boston with David Williams, only a couple of years ago now, about 3 years ago, started using this small RNA technique that Fire and Mello won the Nobel Prize for in 2006, and said, if we can put this small RNA into cells and turn off the BC11A gene, the fetal globin gene will come on. And they've got an extensive clinical trial going on in Boston, and the results are very promising. Now, you need to be a molecular biologist to understand this slide, but this is how small RNAs are made. You have to put in a gene therapy vector into the blood stem cells. It makes this RNA. This RNA binds to the BC11A RNA and stops the BC11A being made. So all that was happening, but something else was happening. CRISPR gene editing had been invented. I won't explain everything about it, but it was a way developed from a bacterial system where we can change individual human genes. And so the first thing people tried is, can we change the sickle cell mutation back? But it turned out that CRISPR works very well in some cells and not others. And just by bad luck, it needs to cut and then repair. And the repair pathways don't work very well in blood stem cells. I wanted to show the pictures of the people who developed CRISPR. I think they will win the Nobel Prize, but they haven't yet. Emmanuel Charpentier, Virginia Sickness, and Jennifer Doudna developed CRISPR. And what they are trying to do, again, how do we stop BC11A? What they're doing here is using CRISPR to disrupt the on switch for BC11A, and that will stop BC11A being able to repress the globin genes. But what was I doing all this time in my lab in Australia? So we were also interested in 1948. Janet Watson had said children have fewer symptoms. Then in 1958, people had found some patients who had really high levels of fetal hemoglobin. It was called hereditary persistence of fetal hemoglobin, a completely benign condition, but these people just had more hemoglobin. And sequencing done by Francis Collin of the Human Genome Project found the mutation was in the actual fetal globin control region. And he worked on these mutations, but he couldn't find out how they worked. But we started working on them. And there's lots of families, and these are just some families trees, of people who have mutations in their fetal globin gene. And the gene is on throughout their life. So Alistair Fennell, who's worked in my lab, but now has gone to John Stam's lab, the son of George Stamatoyannopoulos in Seattle. And Bika Wienert came from Germany, and Gabby Martin is a Sydney PhD student, and they all worked in my lab in this building at UNSW Sydney. And we did a very simple thing. We said those mutations at the beginning of the fetal globin gene, I wonder if a repressor binds. And we said to Gabby, see if you can find out what that repressor is. Test every known DNA binding protein you can. And there's Gabby at the bench beginning to do this. And in the first experiment, now this is an experiment where we have a piece of DNA at the bottom and we add different proteins. And when you add BCL11A, it binds to the beginning of the fetal globin gene. And suddenly everything made sense. Up till this moment, no one knew how BCL11A turned off the fetal globin gene, but now we knew it bound directly to the beginning of the fetal globin gene, sat on it, stopped the gene from coming on. And that was a hugely important result for us. And those mutations that Francis Collins had discovered initially, each of those mutations in those different families, those mutations stop BCL11A binding. So in most people, BCL11A binds to the site. If you make a mutation, you don't see that black band because the mutation has changed the shape of the DNA so that BCL11A no longer binds. And so we said, what if we use CRISPR not to interfere with BCL11A, but just to interfere with the fetal globin gene? Will the fetal globin gene turn on? And that's shown in this very complicated slide. Here's the fetal globin gene. Here's the guides that you use to send CRISPR to the beginning of the gene. And here's the data. Normal cells have very little fetal globin, but if you make those mutations, you get a large amount of fetal globin. And this is a particular experiment that shows just how much you get. So what a simple result. BCL11A binds to this region. And what that means is if you disrupt that using CRISPR, you could alleviate the disease. And so this is being done in France, in the US, and we're talking to a biotech company about how best to do this. But there's more. People kept saying, why work on the fetal globin gene? Why not just repair the sickle cell mutation? And as I explained, we can't do that because you have to cut and repair, and that doesn't work in blood stem cells. And then this wonderful scientist, David Liu at Harvard, I follow David on Twitter, actually. He's one of the best commentators on the coronavirus too. He's well worth following. He invented a new way to change DNA called base editing. And here on the front page of Science, it says new prime editing, which is even better than base editing, could surpass CRISPR. The first base editing could change many, many different letters of DNA into other letters, but it couldn't, just by bad luck again, change the sickle cell mutation. But now David has invented prime editing, which is a mechanism for changing any letters you like in DNA. And I'm very optimistic that in the future, you'll be able to change the sickle cell mutation back in blood stem cells. So with that optimism, you'll probably be sitting there thinking, well, if we can change that DNA, why don't we just do it? And it's important I explain to you that changing humans is really, really hard. And it's hard for two simple reasons. We have trillions of cells. If you want to change a human, you have to change every cell, or at least every blood stem cell, to ensure that the future blood is changed permanently. And the other thing is our cells don't welcome new DNA. Our cells are surrounded by membranes that prevent DNA getting in. And if DNA does get in, and you know what a virus is just a little package of DNA. Excuse me, if DNA gets in, our cells treat that DNA like a possible virus and silence it as fast as they can. And so to get DNA in, you have to take bone marrow out of the patient, grow them in a flask. You have to add the CRISPR base editors into the cells, which is not easy to do. And then you have to check that they've done their job, and then you have to transplant the bone marrow back in. So it involves essentially a bone marrow transplant out of someone and then back in, which is complicated. So is there a better way? So some researchers are saying, is it possible to do in vivo CRISPR base editing? Could we just inject CRISPR into someone's arm and let it find its way around the body and edit the right cells? Now, that sort of experiment, there's evidence that might work in a mouse. You need to have a lot more of it in humans, and no one knows whether that is safe or effective. So that is not available as a therapy at the moment. But one day, I believe it is possible that it could be done safely, but a lot of testing will have to be done first. But there's another idea, which I'm actually most excited about. And it's to say, yes, we can inactivate BCL11A with a small RNA. We can inactivate BCL11A with a CRISPR gene editing molecule. But why don't we just see if we can find a drug that binds to BCL11A and turns it off? So people are trying to do that, but no one's been successful. But a friend of mine who was also a postdoc with me in Harvard in the early 90s, he said, it's hard to make a drug against a DNA binding protein, but I bet the DNA binding protein is regulated by something called a kinase, which is an enzyme that adds phosphate groups to proteins. And he said, there are lots of drugs against kinases. I'm going to search all the kinases and see if any of them are required for BCL11A activity and stability. So Gerd Blobel, he's Gunther Blobel's nephew. Gunther Blobel's a famous molecular biologist who won the Nobel Prize. And Gerd is a younger molecular biologist. Gunther Blobel passed away a couple of years ago. And Gerd works in Philadelphia. And he did this CRISPR-based screen, which just means you use CRISPR to test each kinase at a time. So druggable kinases. And he tested, there's about 1,000 druggable kinases. He tested every one of them. And he found a kinase called HRI, and this is published in Science a couple of years ago, affects BCL11A levels. So now he's working with two very important pharmaceutical companies to develop inhibitors for this kinase. In theory, that could be excellent, but we don't know how well those inhibitors will work, and we don't know what side effects there might be. So we don't know whether or not this could be a cure, but I wanted to explain it to you because I think this work of Gerd's is so important. And if it is a cure, it could come very quickly when it comes. But on the other hand, it may not work at all. It's still being worked on. So I'm getting close to the end now. So we're all still working for other strategies. And I just wanted to show you about here in Australia. So that's my lab, and we always try to put the Sydney Opera House in the background so everyone knows exactly where we are. We took that late last year. And then Andrew Perkins, he also, some of you will know Andrew, he's a clinician scientist, an excellent clinician scientist. He also worked in Harvard with Gerd Blobel and I. We were all in Stu Orkin's lab together. And Andrew's doing excellent work on sickle-cell and related blood disorders. Steve Jane worked in Washington in National Institutes of Health with Art and Ian House, and who was a leader in gene therapy for thalassemias and sickle cell. And Steve now is at Monash, as is Andrew. And John Rasco, who's here in Sydney. John is a pioneer on gene therapy for blood diseases and has been involved in some of the biggest trials in Australia and globally. And he's a very well-known scientist. So there's quite a few of us in Australia who are interested in this problem. And I think the research is excellent. And I'll just go to, this is my second last slide now. So to me as a scientist, sickle cell is the most famous, most abundant and best studied inherited disorder. Generations of researchers have been working towards a cure. And I believe that one day we will have better treatments. In recent years, CRISPR genome editing is meant you can modify cells, but there's still this complicated procedure of having to get the bone marrow out, change it, get it back in. I hope that one day in the future, there will be drugs that turn on fetal globin, and I hope that they can be used in developing countries across the world, as well as in countries like Australia. And perhaps there'll be a drug, or perhaps you might be able to inject CRISPR for in vivo gene editing, although none of us know if that's safe. And I'd also say the health system in Australia, clinician scientists like Andrew, Steve Jane, John Masco, we have some of the best clinician scientists and clinicians in the world. We do not have the scale of research that they have in the US, but we do have the quality, and we collaborate. I collaborate with Gerd and other people. I've got my lab website, which talks about what lab life's like on our blog. I've got Twitter, and I just want to show, the last conference at Oxford, 2018, those are all the researchers who've basically dedicated their life to this problem at Warwick Castle for the dinner. I actually missed that dinner because I had to come back for something, but I did go to the rest of the conference. And that just gives you a picture. These are the people who meet every second year. Okay, it's 8:40 now. I think I'm pretty close to time. Now I have to find out how to stop sharing my screen. It's just up here. So I'll do that now and hand back to you, Agnes.